So hello friends, welcome back again to another lecture series of translation in prokaryotes. In the last lecture series we have already studied about the tRNA charging, and the tRNA charging means that the tRNA is added to the amino acid sequence, and each amino acid sequence is specific to each tRNA. So accordingly the tRNA becomes charged and the initiation phase starts. So now in this phase we will in this initiation phase we will study about the how the initiation takes place. To begin with the process, to begin with the initiation process, we need to understand what are the different components present to form the initiation complex. Okay, there are different forms of initiation complex that is formed. One is the small subunit of the ribosome, and the other is the large subunit of the ribosome. There are two different units of ribosome which is present, and one is known as the small subunit which is 30S, and the other is known as large subunit which is 50S. And this complete set will form the 70S ribosome in case of prokaryotes. So within this ribosome there are present a ribosomal RNA or the rRNA, and also within it is present a protein. There are proteins. So this protein and rRNA together forms the ribosomes. And also we need the mRNA, which have some specific sequences also known as start codon, and they will be having AUG in the start codon, and this is same for both in prokaryotes as well as in eukaryotes. And they also have some stop codon out there in a downstream where we will have sequences like UAA and two more such sequences. So now the formation of this initiation complex is occurred in two ways. First is the formation of 30S initiation complex. It is the most vital stage or the vital part of the initiation complex formation, because the 30S subunit complex formation starts the process of initiation. Okay, and there are different types of enzymes involved in it, enzymes and also different types of factors, and these factors are known as initiation factors. Translation initiation factor, which are associated with this 30S subunit. Okay, 30S subunit as given in the below diagram. This is the 30S subunit. So now these initiation factors are mainly IF1, IF2 and IF3, and within these three different initiation factor, the IF2 is a GTPase protein factor, okay, which can use the guanine triphosphate to hydrolyze, and it will form GDP plus inorganic phosphate, and they will release the energy, and that energy will be used by this ribosome to make the reaction in forward direction. Within this 30S subunit, there are three different regions: the E region, P and the A region. In E is the exit site, as I already mentioned it before, and P is the peptide synthesis site, and A is the amino acid tRNA heading site. So there are three different sites in the 30th subunit, where in the IF3 will come, the IF3 or the initiation factor 3 will come and bind in this E side, and the IF2 and IF1 will come and bind in the A side. So this E and A side will be now blocked. That means they are not free now. So the beginning, this IF3 and IF2 and 1 will come and bind with the E and A side. And see here in the mRNA, this is the mRNA, and you see here that this ribosomes comes and bind in the mRNA, where the ribosomes has a protein part and also a rRNA part, where they will have a sequence in the rRNA. That is, this rRNA is also known as the 16S rRNA, which is very much conserved in all the different species. They does not get mutated. Okay, they remains exactly, the sequence remains exactly from generation to generation, and this rRNA has some specific sequences which is complementary to the mRNA. Okay, so here you can see in the mRNA there is a start codon, and in the upstream of the start codon, nearly 8 base pair upstream, you will find 8 base pair upstream of the start codon, you will find some sequences in the mRNA which is known as the Sine-Dalgarno sequence. This is known as the Sine-Dalgarno sequence, which will be having this type of sequences UCC. And complementary to these sequences, in the 16S rRNA of the ribosome, you will find the sequences as U will be complementary with A, C will be with G, then G, then U will be with A, and C will be again with G. That means in the 16S rRNA, you will find this type of sequences A, G, G, A, G. So before starting the initiation or before starting the elongation part of the translation, and before finding the start codon, the 16S rRNA binds with the Sine-Dalgarno sequence, and then they will provide some signal, and they will provide some signal to the ribosomal rRNA or the ribosome to go and bind with the start codon. They will find different start codon out there, because when you see in the mRNA there are different start codon, not only a single, there are many more start codons there, and they need to bind to a single start codon and bind properly, and they can do the elongation process. So the 16S RNA will go and bind with this Sine-Dalgarno sequence, and they will send a signal to the ribosomal RNA or the ribosomes to go and bind at the start site, where they will start the initiation process. So in the next step you will see here that as soon as the start codon is found here, the formilated methionine will come and bind with the tRNA. This is the tRNA and this is the formilated methionine. So this is the formilated methionine and this is the tRNA. Okay, this tRNA will be charged with the formilated methionine, which is a amino acid. This is a amino acid. Okay, so this is a amino acid and they will bind with this tRNA, and now they can, this tRNA can now find the start codon and they can form the initiation complex, RTS initiation complex. So as soon as they formed a 30S initiation complex. Now, in the next step, they need to form the 70S initiation complex. So, for forming the 70S initiation complex, this the large subunit of the ribosome, that is 50S, need to come and bind with the small subunit out here. Okay, they will come and bind here, the large subunit, and the initiation factor will now get released. And with the release of this initiation factor, the GTP will get hydrolyzed and they will provide the energy with the release of GDP plus inorganic phosphate. Now you see in the next step, the 70S initiation complex formation starts to begin with the addition of tRNA and finding the start codon out here. They will now form the initiation complex and they can now begin the elongation process. And there are some other binding site in the large subunit of the ribosome. I forgot to mention it before that there are some binding site which is known as translation factors binding site, which will be studying in the next video lecture. But now what happens after the binding of this large subunit here with the small subunit? Now this tRNA, and they can now bind with the start codon here, and the E side and the A side is now free. So now in the next step, during the elongation part, the next tRNA will come and bind with the A side, where they will be charged with another amino acid, but this will not be formilated one. So now this A side will be blocked like that, and this whole unit of this ribosome will move one codon in the downstream. So as soon as they move one codon towards the downstream, this E side will now go, this P side will now go to the E side, and the A side will now be the P side, and again the A side will be free, and here another tRNA will come, another charged tRNA will come and bind with the specific sequences present in the mRNA. So these were the initiation process ends here, and in the next video we'll come with the formation of the elongation process, and also we'll see how the termination takes place in case of translation in prokaryotes. So I hope this video will helpful to you. If you like my video, do subscribe and also do share with your friends. Thank you.